Okay, once again we are back with the mammalian histology. We are in TYBSc Zoology class semester 1 paper 2 mammalian histology. We are discussing about the uh, point in the syllabus that is 3.5 topic from the syllabus. We are discussing glands associated with digestive system. In the previous lecture, previous part of the salivary gland part 1, we have seen basic histology of all the different types of glands. Now we in this slide we will focus specifically on the different types of glands present in a salivary glands. In salivary glands you will find there are parotid, sublingual and submandibular. These are the primary major glands present in a salivary gland. So we will see that in this presentation. Okay, so in this picture you will see exact arrangement of a submandibular, parotid and sublingual gland. So brown color portions represent the salivary gland all together. All these brown color area are the salivary glands. Here just above the ear portions on the cheek above the major mandibular bone, you will find these are the parotid ducts. So they are the parotid duct and these are the parotid glands. So all these are the parotid glands and this is a parotid duct opening that secretion pouring into the jaw. Now above this major jaw bone, this brown color structure represents the submandibular gland, and there is a duct opening that submandibular duct opening the secretions below to the tongue, and just located just below the tongue, these are all sublingual glands. Okay, and they have their duct to pour their secretions, minor and major sublingual glands. Lingual words, that means below the tongue. Mandibular means jawbone, mandible bone, and parotid, that is related with the bones situated just above to the ear portion. So parotid glands, submandibular glands, and these are the sublingual glands. All these are the salivary glands. Basically they are salivary glands and these are the part of the salivary glands pouring their salivary secretions in the mouth. Now, their location is completely outside the alimentary tract, though they are associated with the digestive system, but they are not located inside the digestive tract. But their location is altogether outside the digestive tract, and through the ducts they are connected with the alimentary canal. So here you can see lot of blood. These ducts are opening, this opening and pouring their secretion in the mouth, and that way they are connected with the digestive system. Then all these salivary glands are compound alveolar or tubulo-alveolar in type. Already we have discussed what is that compound alveolar, what is that compound gland is, what is alveolar gland is, what is tubulo-alveolar gland is. Okay, compound means they have a lot of branchings, networks of a duct. Alveolar, they are grape like a small capsule like structure is there, and tubulo-alveolar means a large long tube like structures and alveolus like structure combined together, and both the sections tube as well as alveolar portion is a secretory. Then here they are the parotid, submandibular and sublingual glands in a salivary. Okay, but along with that some minor glands are also present. Minor salivary glands are also present in a jaw portions. So they are at the lip, cheek and soft palate on the tongue. You will find these minor glands, salivary glands. The functions of salivary gland is to secrete saliva composed of what? Water, mucus, some proteins, salivary enzymes, that is amylase, and lot of immunoglobulins are there, and the lactoperoxidase. We are very much familiar with the amylase enzyme in a saliva, so that is the source of salivary gland. Then what else is? The water is there, mucus is there, and proteins and salt. They serves to moisten the food. Dry piece of chapati, if you go on continue chewing, that get partially digested in mouth only because of these enzymes, as well as it is get moistures and chew because of water content produced from the saliva. Then they lubricates and moisten the oral mucosa and lip. You cannot imagine your mouth become very dry. Okay, so it should be always full of moisture, and that is the function of salivary gland. They initiate the digestion of carbohydrates. As we said that salivary amylase, this is a primary enzyme for initiation of a carbohydrate digestion. Now, basic organization of this salivary gland is they have this opening. This is a mouth portion, mouth internal mouth surface. They have this main excretory duct, anterior lobular duct, then lobular duct, then the main excretory duct. So all this is a network of duct bringing secretion from acinus to the surface in the mouth. So this is a chain of duct pouring secretion on the surface in a mouth. Now this yellow portion indicate a group of many acinus, and that group of many acinus is now called as a lobe. And in that smaller lobe you will see that is called as a lobules. In that smaller lobules you will see many such acini, many such small alveoli acini. Okay, alveolar acini. So all these small alveolar acinus come together to form the lobules. All lobules come together to form a large lobe, and that lobe come together to form that major portion of that gland. Here you can see histology of tube system. Histology of tube system which pour the secretion outside. So these are the cells of striated duct. This, okay, they are the cells of striated duct. Their function is contraction. Striated word related with the muscular contraction. Now this is a histology of intercalated duct, intercalated between the two acini. Okay, so here all acini are connected with the intercalated duct. So histology of intercalated ducts look like this. Here they are simple cuboidal cells, but they are also secretory in function. Some less amount of secretion happens there. Then striated ducts, here you will see striations because they have role in a contractions. Otherwise the secretion cannot propelled forward. So for contraction purpose they are striated. Then these are interlobular duct, and this is a main excretory duct where you will find a lot of epithelial cells, but compound epithelial cells you see here. Here there is a pseudo compound epithelial cells. Here the columnar cells, epithelial cells. Here the cuboidal cells. Okay, now if we see the structure of single acini, what here it's a simple basal lamella or basement membrane is there. On the basement membrane there is arrangement of acinar cells. That acinar cells they are responsible for the secretion. So their nucleus is arranged at the periphery, and all these acina is externally supported with the myoepithelial cells. Myoepithelial cells are muscle cells which are supporting out from outside this acina, and the role of myoepithelial cell is to contract, so the internal secretion get propelled forward in the intercalated ducts and enter into the striated duct. Then striated duct contract and they release their secretion outside. Okay, now here different types of acini we see in a salivary gland. Uh, Based on the type of secretions, they have separated here on the mucus secreting, serous secreting, and mucus as well as serous secreting acini. Basic structure is same. There is a basal lamella, over that there is epithelial cells and surrounded with the myoepithelial cells. So here again you see in every kind there is a myoepithelial cells, but in case of here mixed type of acini, you will find the serous producing cells are arranged outside the this mucus acini. Mucus acini, and some portion of mucus acini is supported with the serous producing gland. Here it is a mucus producing gland, mucus producing acini. Here is a serous producing acini, and here mucus producing acini supported with the serous producing cells, and that are called as a serous serous dermalion along with the myoepithelial cells. Now here under electromicrograph you will see a different detailed structures of the serous and mucus producing cells. Under light microscope it look like this. It is full of mucus, and here it is some stain with some zymogen granules, here secretory granules, some nucleus, and some striations indicating the cell organelles. But under electron micrograph, you can clearly reveal the presence of pre-ribosomes, then mitochondria, endoplasmic reticulum, Golgi complex, and lot of secretory granules accumulated at the apical portion of the serous cells. Whereas in case of mucal cells, you will find here accumulation of that mucus cells here, mucus vesicles, and very few amount of Golgi complex, endoplasmic reticulums, and some mitochondria. So serous producing cells organelles are more functional as compared to the mucus cell producing cell organelles. Now if in university exam they ask about the difference between serous and mucus acini, or if we try to learn these two in comparatively, it will be easy for understanding. So serous acini are compound alveolar. Remember that compound alveolar, or recall from the earlier lecture, compound alveolar. But mucous acini are compound tubular, or sometimes a tubular alveolar glands. Okay. So they are simple compound alveolar, so they are small in size and round in shape. As they are compound tubular or tubulo-alveolar, they are large in size and occupy a lot of space, and they come in a many shapes. That bunch structure goes on increasing. Here in a serous acini may shows very small lumen, or mostly the lumen is obliterated, not seen, because the way of releasing the serous is bursting of cell. So that secretion is remains stored in a apical portion of cell, so they don't require the internal gap or internal lumen. But in case of mucus, that secretion is a merocrine kind of a secretion, so continuously the mucus is poured into the lumen, and there is a gap between the acini to store that mucus. Here they don't store that secretion in a hollow cavities, but they store that secretion in a cell only. So there is no lumen you see in a serous. Here you will find quite good size of a lumen. Here the serous acinus is lined with small pyramidal cells, and their number within an acinus is more. Serous acini is lined with the small pyramidal cells, and their number within acini is more. Here mucus acini is lined with relatively large size of columnar or pyramidal cells, and few cells line an acinus. That is a shape of cell they have compared here. Serous acini stain dark. We already seen that hematoxylin color this serous acini in dark color. In mucus, that He stain do not stain these mucus cells, so it is light in color. The serous cells may be present as demulins on the mucous acini. Earlier slide only we have seen that mixed type of a gland. You will find some serous cells located on the periphery of mucous acini, where you will not see any such mucous cells present on the serous acini, around the serous acini. Here, serous cells are mostly pyramidal in shapes. They are small in size. Obviously, mucous cells are short, columnar to pyramidal in shape. They are large in size compared to the serous cells. Continuously mucus secretion is needed, and so the size is larger. When stained with fluorescence, this is dark stain. Here the light stain nuclei are rounded and placed near the center, but more toward the basal part of the cell. Right here, the nucleus is flat and arranged at the periphery. Both nucleus are arranged periphery, but here it is a round nucleus. Here it is a flat. The mucus secretion compress that nucleus. Here the apical portion is filled with the secretory granules, zymogen granules, but obvious that serous is packed into the zymogen vesicles only. Here the apical portions contain large number of secretory granules. Mucinogen granules are packed there. By staining by using specific stain, you can reveal the presence of zymogen or mucinogen granule. The base of serous cells is basophilic, blue in color, and apical portion is acidophilic, that is pink in color. Here cytoplasm takes light pale stain and looks empty or vacuolated. This because of the mucus is lost the preparation of hematoxylin stain slide. When during the microtomy, when we prepare the slide, that time much of this mucus get dissolved, and so it forms a empty space, and so that cell don't get hematoxylin stain, eosin and hematoxylin stain. But here that serous secretion don't get dissolved and it get good stain. Okay, so here is a parotid gland.